Okay, multitasking here. So chapter 25 is the first chapter in unit 2. And when it comes to naming amines, there's several different styles that are in use. And we'll concentrate on the chem abstract style. So the chem abstract style is like, it's an American Chemical Society style that is used in all their publications. So again, the root is the longest chain that's bonded to the nitrogen. Leave out the E, terminate in amine, and then the amino group, the NH3, NH2 I should say, gets the lowest possible number. Okay. So if you look at 1-propanamine, what does that mean? That means propane with an amino group on the number 1 carbon. But similar to what we did with alcohols, you can also say propane 1-amine. And so if you have an alkyl group and then the amino group is attached to that alkyl group, it's called an N-alkyl. I think I said that wrong. If you have an alkyl group attached to the nitrogen, it's called N-alkyl, such as N-ethyl, N-diethyl. Then you try to put everything in alphabetical order. And so the chem abstract names are not in brackets. So here's the chem abstract's name. Here's the IUPAC naming system, which is not always used. Then here's the common naming system, which unfortunately they still use a lot in textbooks and everything else. So if we analyze this structure right here, and again, my goal is to get it so you have a name that describes that particular structure and no other structure. And you do the best you can with the grammar. Minor grammar mistakes, not important. So you have propane with an amino group on carbon number 1. It could have been on carbon number 2. If it was, it'd be called 2-propanamine. IUPAC, simply you call this the amino group. So 1-aminopropane. And the old-fashioned name, here is the alkyl group, and here is the name of the functional group. So N-propylamine. And even in that system, most of the time, the functional group and the name of the alkyl group are 2 separate words, but for some reason for amines, they tied it all together. So here is the longest continuous chain is propane. So this is propanamine. Where is the amino group on carbon number 1? And that covers this whole part, but it doesn't cover this methyl group up here. So how do we say that? If the alkyl group was on here, we'd give it a number. But it's not on a carbon, it's on the nitrogen. So we just say N-methyl 1-propanamine. Notice that the IUPAC is very similar, N-methyl 1-aminopropane. And if you look at the old-fashioned name, the common system, this would be the propyl group. This is the methyl group. So it's propylmethylamine. Notice that they did not use alphabetization. Sorry. So this one is similar. So you got butane, and the nitrogen is on the number 2, so it's 2-butanamine. This time you have substituents, 2 substituents on the nitrogen. And just like with, if they're on carbon, on the same carbon, you have to give the number twice. So this is NN-dimethyl 2-butanamine. If I saw the word N-dimethyl 2-butanamine, I wouldn't know how to draw it. I couldn't trust that name. Then we get to this one, and the root of this is aniline. Okay, so that's aniline. Now you're going to have to remember the name of aniline. So this would be carbon number 1, carbon number 2, carbon number 3. So on carbon number 3, there's an alkyl group. That alkyl group has 1, 2, 3, 4 total So it's a butyl. Which butyl is it? You see it split on the end like a fishtail. That's iso. So 3-isobutyl aniline. Okay. Let's see. So look at this one. I see in here there's aniline. So what is substituted onto the aniline? Well, here's a 1, 2, 3. So here's a propyl group, right? So it's carbon number 1, 2, 3, 4. So it's 4 propyl aniline would cover all of this. And then what's on the nitrogen? We have a methyl and a propyl. So we have to say N-methyl, N-propyl. Do not say N-N-methylpropyl because that would mean something different. Okay, so every substituent gets an N or a number if it's on a carbon. Okay. So does anybody have any questions so far? Because I know that was a little bit complex, but it's based upon other naming systems as well. Any questions? And if you look at how we name this, it really has to do a lot with how did we synthesize this compound? You start with benzene, then you would put the amino group on there. The amino group is orthoparo-directing, so you end up having, in this case, on the para position. And then you came along later and put the substituents onto the nitrogen. And then what about diamines and triamines? So we just do a similar way as we did with diols and triols. So this would be 7 in a row. So it's heptane diamine. And where are the amino groups? Number 1 and number 7. You can also do it that way. 1, 2, 3, butane triamine. Okay. You see, whichever method you do, as long as this describes that chemical and no other chemical, that's fine. And then amines react with acids to give salts. And so how do we name these salts? Well, the amine or the amino group simply becomes ammonium. So here we have NN. Wait, why is that? Oh yeah, I don't have a triethyl. It's diethyl. That's supposed to be a 2. I never noticed that before. Let's see. Okay. Maybe I should just, yeah, I should just change the name. Sorry. You see how the naming system makes it so that you can actually see these things? Yeah, okay, that's better now. So this is like ammonia. So ammonia would be NH3. This is N with 3 ethyl groups attached to it. So that non-binding electron pair right there is what makes it act as a base. So here comes a strong acid, and it transfers the hydrogen ion to that non-binding electron pair, which now becomes a bonding electron pair as it binds to the hydrogen. But now, just like the ammonium ion, this has 1 proton more than it does electrons. Okay? So it has a positive charge. And then the chlorine becomes chloride, which is the counter ion. I don't know how, nobody ever corrected me on this. I guess I just really had bad students until you folks. Okay. So here would be the complete Lewis structure for this. So here is the, wait, what the heck am I doing here? Oh no, this is a different structure. I'm sorry, this is this one. Okay, so a lot of drugs are amines. And as you'll see in a minute, amines kind of smell bad, which means that they taste bad. And so what oftentimes pharmacists will do was treat them with an acid to make the acid form. So this becomes very soluble in water compared to this. And not only that, it doesn't smell. So here's a different one. I showed the exact, how do you say, structure of it. So notice that I didn't put the charge directly on the nitrogen. I just put it up here. And there's 3 of these because phosphate has a charge of -3. This is a charge of +1. So look at the name of this. All we did is change the word amine to ammonium. See how that works? Very simple. Okay. So amines are analogs of ammonia. Here's ammonia. Here's a primary amine, a secondary amine, a tertiary amine, and a quaternary ammonium. Because just like with ammonia, there's 4 things bonded to the nitrogen, so it gets a formal charge of +1. Okay. So the physical properties of amines, the bonds in amines allow for dipolar attraction and hydrogen bonding. The bond between carbon and nitrogen is slightly polar. The bond between nitrogen and hydrogen is extremely polar. And you should be able to, again, draw hydrogen bonds between amines and between amines and water. So I'm going to draw some hydrogen bonds here. So let's see. So again, that would be ammonia, right? So if I make this into a methyl group, now that's called methylamine or aminomethane. So I'm looking at the charges here. This would be the partial positive. This would be the partial negative. This would be also a partial positive, and that would be its partial negative again. Okay. So that partial negative would attract the partial positive of another amine. Well, what would that be? That would be the hydrogen. So what kind of attraction would this be? So that covers Londons. All molecules have Londons. This is a dipole right here and a dipole right here or here. So it does meet the criteria for dipole-dipole. Doesn't meet the criteria for hydrogen bonding. Well, here's the NH of one. What do I need in the other one? An F, an O, or an N, and there's the N. Okay, so that is a hydrogen bond. Then this partial positive would attract the partial negative of another molecule. That would be the electron cloud on the nitrogen. So there's another molecule. And again, it's a hydrogen bond because here's the NH of one, and here's the N of the other. Similar down here, this would attract the partial negative, which would be the electron clouds on the nitrogen. So in effect, we're not saying that a solution of methylamine is going to have 3 hydrogen bonds per molecule. We're saying this is the maximum number. It's probably more like 1 or 1.3 or something like that, depending upon the temperature. Nevertheless, they're attracted to each other, so they would have fairly strong attractive forces. So if I look at this one right here, this NH, that would be the partial positive. That would attract the partial negative of a water molecule. And is that a hydrogen bond? Here's the NH of 1. Here's the O of a different molecule. So yes, that is a hydrogen bond. So I could replace all of these with hydrogen bonds. So a primary amine, such as this, could theoretically have 3 hydrogen bonds with water. So what if now I made this into a secondary amine? So for example, that hydrogen now becomes another methyl group. Well, now there's no partial charge on there. So now there's only 2 possible hydrogen bonds between itself and another one like itself, and only 2 possible hydrogen bonds with water. What if I make this into a tertiary amine? Now this last hydrogen becomes a methyl group and there is no partial charge on it. Okay. So, can there still be dipole to dipole attractions between these molecules? The answer is yes. Like if I go over here and I make this a methyl, so this NH over here could still be attracted to this partial negative of the electron clouds. So what if I now make this into a tertiary amine? Well, now we don't have this anymore. So what does that mean? They're both polar molecules, so there's dipole-dipole attractions, but there is no hydrogen bonding between tertiary amines. What about between a tertiary amine and a water molecule? Let's see. So here's a tertiary amine. Here's still the partial negative that's going to attract the partial positive of a water molecule. Oops. So is that a hydrogen bond? Here's the OH of one molecule. All I need in the other is an F and N or an O, and that is a hydrogen bond. So a tertiary amine cannot hydrogen bond to other tertiary amines, but it can hydrogen bond to water. But you can see that a primary amine is going to be more soluble than a secondary amine, which is more soluble than a tertiary amine in water. And it's all because of, how many hydrogen bonds can you actually draw? So I know you must have questions, because that's one of the most difficult things to do, is to draw hydrogen bonds between molecules. Any questions? So that really means that all of you are geniuses, or you're so overwhelmed you have no idea what question to ask.
More overwhelmed.
Yeah, it's a hard thing, like I said. What about the quaternary ammonium salts? Now, they are ionic substances, so they're more likely to dissolve in water. They're also more likely to be a solid because the attractive forces between them are ionic bonds. Okay? So the smell of fish is really due to trimethylamine. Soaking fish in lemon oil or adding lemon juice will convert much of that smelly tertiamine into its quaternary salt and reduce the fishy smell. So, yeah, that's what lemons do with cooking. And here is triethylamine, but it's usually trimethylamine that gives that fishy smell. So what about the chemical properties of amines? Well, they're weak bases similar to ammonia. The reason why they're weak bases, just like ammonia, is because of that non-binding electron pair right there with this partial negative charge on it. So water can act like a weak base, a weak acid. If you put a weak base into water, it makes water act like a weak acid. So in this case, this proton right here left its electron behind and went on to here. So this non-binding electron pair right here became that bonding electron pair. So this now has one proton more than it does electrons, and this has one more electron than it does protons. So those two bonding electrons now became those two non-bonding electrons. So that's the acid-base reaction. And so we can take the equilibrium constant for this equilibrium reaction, which is simply always products over reactants, right? Leave out water because water is a pure liquid. So when I do this, most of these give a constant of about 10 to the minus fourth. So here's ethylamine, has an equilibrium constant of that. pKb is about 3.2. That makes the pKa 10.8 because of this relationship right here. Okay. So I don't know what you know about this, but if the pKa is 10, if you made a solution that was 50% the base form and 50% the acid form, the pH would be 10.8. Okay. So what would it look like to match the blood plasma pH, which was somewhere around 7.4? So a 50-50 mixture of the acid form and the base form would give you a pH of 10.8. What would I have to do to get that pH to go down to 7.4? I would have to add acid, wouldn't I? Okay. So what does that mean? If I'm adding acid to this equilibrium, I'm converting much of this into this because I'm getting rid of this. Okay. So I put this in a red box, which means that you don't have to know how to do this or anything. So what I did here is I solved for this right here, right? Took the negative log of it, which gives you the POH, right? So here's the POH. Here's the pH at that time would be whatever. I can't see that anymore. So essentially what it means is that if you go through the math over here, at pH 7.4, the ratio of protonated to unprotonated would be about 25,000 to 1. What does that mean? That means if you take an amine and you put it in water, it makes it basic. But when that amine pH is adjusted to physiological pH, it's almost entirely in the protonated form. And that's important to know. I mean, if you care about biology at all. So amines are often called a solubility switch. So oftentimes something like this would be insoluble in water, but when I protonate it with an acid, it becomes a soluble ammonium salt. This is the salt right here, right? Cation, anion. Okay. So let me see if I can find this. I even forget what it is. So this is a typical thing that we do in the organic chemistry lab. Say you have a mixture of two compounds and you want to separate them. Okay. So what you do is you dissolve them in diethyl ether, which is really good at dissolving a little bit of polar and a little bit of non-polar solvents. And then you add some water with hydrochloric acid in it, some acidic water. And what you find out that the ether and the water separate out. They don't dissolve in one another. So ether, which is rather non-polar, is going to contain all the cyclohexanol. This will preferentially dissolve in the ether, and this, especially when it's been protonated, wants to dissolve in the aqueous layer. Okay? So then, we use a separatory funnel. So here would be the ether, here would be the aqueous phase. So now we have one molecule in here, and the amine is in here. And we can separate them out. So I do that separation, I get the aqueous layer, then I add some sodium hydroxide and some water to neutralize it. So I'm turning this ammonium back into the amine, into aniline. Then I put in the ether layer and I shake it up and separate it out again. And what I'm going to find is that in the ether layer now is my aniline. And what I have in the aqueous layer is the result of neutralizing the acid that we had put in there. And you can just evaporate the ether. And now you've separated your aniline from your cyclohexanol. Okay. And then here's also something a little bit interesting. If you look at this salt right here, that salt has no nitrogen, has no hydrogen anywhere. So we say that that is not an acidic salt. But if you look at this one right here, that is an acidic salt. That hydrogen right there in blue could potentially act as an acid. Same thing over here. Okay. So, questions so far? You understand why it's a solubility switch? Then here's one that's important biologically, nitrosamine formation. So if I take an amine, as long as it has a hydrogen, that means it has to be either a primary amine or a secondary amine. A tertiary amine wouldn't have that. And I mix it with nitrous acid, not nitric, but nitrous acid. Notice how with the blue I'm going to make water. Now that nitrogen needs another bond, so does that one. So I put them together, and I get what's called a nitrosamine. This is the nitroso group, and it's on the amine. So it's called an N-nitrosoamine. So we don't have to name these, but This would be called N-nitroso, N-methyl methanamine. So the problem with this is N-nitrosamines are known carcinogens of the stomach and the intestines. They're formed under the acidic conditions of the stomach, which produces hydrochloric acid. And so if you have nitrite in your food, here's nitrite, here's the hydrochloric acid that your stomach produces in order to help you digest your proteins. Look what happens. I can form nitrous acid. It's the same nitrous acid as up here. Okay. So what does that mean? Sodium nitrite is put into so many foods. For example, any kind of preserved meats like luncheon meats, hot dogs, some other things. And the reason why it's put in there is because it's bacteriostatic. Sodium nitrite prevents most bacteria from growing. If they're not growing, that means that they're not producing toxins. So you're protecting people from food poisoning. And I ask this question all the time, and I'm amazed that I find people who've never had food poisoning. I've had food poisoning several times. Once right here at the university, back when I was young and I was surfing the North Shore all the time. I mean, I could be held underwater by a wave for, 20, 30 minutes, no problem. Then I was eating at the cafeteria and I started getting sick and I'm trying to get to my car to go home and I couldn't even make it across the street to get to my car. Just laying on the grass, barfing my guts out, thought I was going to die. So any medically fragile person could easily die. And none of you have food poisoning from Clostridium, because if you did, you wouldn't be here. So the reason why the FDA requires nitrite to put into food is because it saves lives. But on the other hand, it does increase your risk to stomach and intestinal cancer slightly. So what can you do to get the protection from the sodium nitrite, but not really put yourself at risk for other cancers? Well, you can remove nitrite by just washing the meats with water. Sodium nitrite is extremely water soluble. Maybe your luncheon meats won't taste as good, but you can add salt back to them. Or you can cook in the presence of oxygen. Matter of fact, I don't know how you cook in the absence of oxygen. That would even be more dangerous. So what that does is it converts much of the nitrite into nitrate, and the nitrates will not form nitrosamines. So where my kids were young, and then there'd be like 15 kids at the house, all playing in the backyard, and now they're all hungry. So you got to make hot dogs or something. I would take the hot dogs and slice them several times, boil them in water, dump out the water, reboil them again, just to remove as much of the nitrites as I could. Okay. So questions so far? So because the neurotransmitters in so many drugs are actually amines, I decided to go into it here. So neurotransmitters and their agonists and antagonists. So the binding of a drug to its receptor protein can be described using this equilibrium right here. Okay, what's the equilibrium? We can use the equilibrium constant, products over reactants. So we call this a binding constant, even though it's just an equilibrium constant. So if you have a large binding constant, what does that mean? You have a lot of the drug bound to the receptor protein compared to what's not bound to the receptor protein. So that's extremely easy to think of. Unfortunately, most of the people, when they talk about this, they use the opposite. They talk about the dissociation constant rather than the binding constant. And it's simply the reverse of the binding constant. And it's kind of unfortunate that they do, but they do. So in this case, a small dissociation constant represents strong binding of the drug to the receptor. So if you ever take pharmacology or study it, you better be sure what you're talking about. Are you talking about KB or KD? Because they are the opposite direction, right? One is the inverse of the other. So regardless of what you're looking at, an agonist is a molecule that binds to the receptor protein and triggers a response. So if a drug or a neurotransmitter binds to the receptor protein and triggers a biological response, that means it's an agonist. It's going to have a small KD or large KB in order to bind to the receptor protein. What is an antagonist? An antagonist blocks the action of the agonist by binding to the receptor protein more strongly. So it's going to have a smaller KD, binds to the receptor protein, and outcompetes the agonist. So for example, histamine is an agonist. An antihistamine can also be considered an agonist. Wouldn't you expect that if the histamine binds to the receptor protein, an antihistamine is going to have to resemble it in structure fairly well? So you want the antihistamine to act as an agonist, to bind to the receptor more strongly and outcompete the agonist. So let's just keep those two words in mind right now. Say you have allergies and you take antihistamines. When is the best time to take the antihistamine? Would it be after you've had all the effects of the histamine? Or would it be maybe before as a prophylactic? If you take it before, the antihistamine or the antagonist binds to the receptor protein, preventing the histamine, the agonist, from getting there and causing the biological effect. So yes, antihistamines should be taken prophylactically. So an inverse agonist binds to the receptor protein and causes a response, a biological response that's opposite to the agonist. A co-agonist is when more than one agonist is required to trigger the biological response. An irreversible agonist permanently and covalently binds to a receptor protein. It has an extremely small KD. In other words, it basically takes the receptor protein out of action permanently. And a paradoxical effect is when a drug gives the opposite effect than what is expected. And I'm sure you've probably know of examples of this. So maybe a student starts to drink a whole bunch of coffee so he can stay awake and study. And then you just drink a bunch of coffee and all of a sudden you like fall asleep. So the caffeine is doing the opposite effect of what you would expect. It's a paradox. Well, the reason why that happens is because the central nervous system, caffeine is a central nervous system stimulant. And what did it do? it stimulated your central nervous system to the point of exhaustion. So you need to go to sleep. My youngest son was extremely hyperactive. So he was so hyperactive, I couldn't even put him into school. It just wouldn't work. I took him to all kinds of different alternative views, natural paths. I had people with crystals, like putting crystals over him like this and prescribing these special, herbal stuff, and just nothing worked. I resisted giving him Ritalin or anything else like that, but when I finally decided there was no choice, when I gave him Ritalin, or if I gave him Dexedrine or something like that, at low amounts, it calmed him down. Regular people, it would, hype them up. That was the paradoxical effect, and that's when I knew that's what I had to do so my son could go to school. But these right here, an agonist and antagonist, you should really know what those are. And be careful with what are they talking about and what you're reading. Is it KB or is it KD? Because they're opposite. So dose response curves are used to determine the potency or efficacy of a drug. And so here is blue curve, green curve. I didn't say which one is which. But say this green curve right here is the agonist. Okay. So this point of inflection right here is the potency and the efficacy based upon the concentration that you have to use. Then you invented a drug that's going to compete with this natural agonist. And here's the dose curve for the drug. So what do you think? Is this blue one going to be able to outcompete the green one? What does that mean? The blue one is 50% bound at maybe 0.08. This is nanomoles per liter. The green one, 50% bound to the receptor at, what is that? Oh, it's logarithmic. So that's maybe, I don't know, 6. So this one would be an effective drug. It could outcompete the natural agonist. If they were reversed, would it work? Say, for example, here's histamine binding to your histamine receptors. Here's an antihistamine. Is that antihistamine going to be effective? The answer is no. You would have to flood your body with it in order to get any effect at all. So drugs in general could be more effective if they're taken at the time when the proteins that they target are the most reactive. So they have looked at close to 20,000 genes to see, like during a 24-hour period, when are these genes the most active? And they found that quite a few of them do cycle up and down during a 24-hour period. And almost 1,000 of them are those that are actually known drug targets. So we keep looking more and more about how drugs work and how their targets work, so that eventually we'll be able to get the most effect out of the smallest amount of drug necessary, because all drugs have side effects. So according to this scientist at Pfizer, to get a single drug to patients requires 12 years, 36 clinical trials, and 8,500 patient volunteers. And only 3% of cancer drugs tested in clinical trials been approved to treat patients. Yeah. So it is no cheap and easy way to get a drug to market. And then what happens as soon as you get a drug to market? What do you see on TV and radio and Instagram and everything else? All these lawyers fishing around for class action lawsuits. Oh, this drug is known to cause cancer. This drug is known to do this. So those lawyers who do the class action lawsuits have the potential to make millions, if not billions. Okay. So in 2018, 22 women were awarded $4.6 billion after claiming that talcum powder caused ovarian cancer. I'm like, what the heck are you doing with the talcum powder? And talcum powder is an inert substance. And the similar type of thing has been going on with, what's the word? That herbicide, glyphosate. But even the FDA claims that glyphosate is not a carcinogen. It doesn't matter. All you have to do is convince a jury, and it's going to make you billions. So one way to categorize drugs and agonists and antagonists is with the LD50. That stands for the medium lethal dose, causing 50% mortality. Okay. So anyway, most of these studies are done on test animals. You're not allowed to do them on Republicans or anything. It's against the law. So if you look at botulism, which is also the same compound used in Botox treatment, is probably the most lethal substance known. So a friend of mine was working with the botulism toxin, and the precautions that they had to use was actually really quite incredible, but it's extremely toxic. So botulism toxin comes from the bacteria, right? Rattlesnake venom, curare, the black widow spider, hemlock, codeine, LSD, nicotine, cocaine. Why is nicotine here twice? I don't know. By the way, nicotine was probably the very first thing that they tried to use to eradicate the coqui frog in Hawaii. So they sprayed it into the jungle, hoping that it would get onto the coqui frog, but apparently it would not. But if it does get on the coqui frog, it's absorbed by the skin, kills the frog. So you can see that some of these that are in normal use by drug addicts and even non-drug addicts are fairly potent. So here's Roundup way down here. Fructose is more deadly than Roundup. And what's even more deadly is sodium chloride. But it's amazing. Even blood sugar you can get LD50 for. So neurotransmitters are categorized as either catecholamines or non-catecholamines. Catecholamines contain 1,2-dihydroxybenzene in the structure. So here's 1,2-dihydroxybenzene. Okay, this is the natural agonist dopamine. Here's another catecholamine, the natural agonist adrenaline. And here's the precursor to adrenaline. So all of these are catecholamines. See that? Okay. So So dopamine is a primary amine, adrenaline is a secondary amine, and norepinephrine is also a primary amine. So what's the difference between norepinephrine and epinephrine? Let's see, carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon. Oh, there's an enzyme that takes norepinephrine and puts a methyl group on it, an N-methyl. So norepinephrine really is N-methyl adrenaline. And then we have non-catecholamine neurotransmitters that don't contain 1,2-hydroxybenzene. So here's the first one we looked at is acetylcholine. It's a neurotransmitter. Okay. So right now we can look at acetylcholine and look at that nitrogen right there. It has 1, 2, 3, 4 carbon groups attached to it. That's why it has that positive charge. It's a quaternary ammonium molecule. Later on, we're going to see that this is an ester linkage right here. And this part came from a carboxylic acid, and this part came from an alcohol. When acetylcholine neurotransmitter is destroyed after its use in the synapse, this ester linkage is hydrolyzed right here. Another non-catecholamine neurotransmitter is glutamate. So glutamate is an amino acid. Here's the amino group. Here's the carboxylic acid group. Okay. And here's GABA. GABA is called gamma aminobutyrate. So it's an old-fashioned word for a carboxylic acid. Now we would call it butanoic acid. So here's a carboxylic acid. So here's an acid. Here's an amino group. Oh my God, that's an amino acid. Here's serotonin. You can see it's a primary amine. Over here, it's a secondary amine. And it doesn't have 2 alcohol groups, otherwise it would be called a catecholamine. So do you need to memorize any of these structures? No, you do not. You simply need to be able to name these types of amines over here, right? Stuff like this. That's what you need to know how to do. So what are alkaloids? Alkaloids are amines that are derived from plants. And there's 4 categories, and many of them really have profound biological effects. So one is the narcotic analgesics. And what they do is they relieve pain, induce sleep. Those would include opiates from the opium poppy. If you think about it, relieve pain and induce sleep. Well, that's kind of like euphoria, isn't it? So opium actually contains both morphine and codeine. So use of opiates and other addictive drugs cause an increase in demand for the opiate receptor. So now how would your cells respond to that? Your cells need these opiate receptors so they can perform their normal everyday functions that they were supposed to do in your body. But now every time you put a receptor out there, it gets taken over and taken out of action by a narcotic. Then eventually you're going to say, I got to make more of these receptors. So the genes for the opiate receptors are then upregulated and overexpressed. So now when you take the drug away, that cell has too many opiate receptors. And downregulation is difficult. It takes a long time. And this is a biochemical basis or evidence for addiction. So all these people that try to tell you that addiction is all mental, they're full of something. So here is morphine, and here is codeine. What's the difference? Simply, again, I took this secondary alcohol right here, and I made an ether linkage out of it. So this is methoxy morphine. That's what codeine is. So what about pain? People study pain. So most animals and insects avoid wasabi because it causes pain. I guess some people might not know what wasabi is. Is that green mustard stuff that we put on certain foods? I guess it's wasabi sounds like a Japanese name, not a Chinese name, but I love wasabi. And this is a fun word to say as well. But low dosages give you that zing that we like from wasabi. But here's the active ingredient. So allyl isothiocyanate denatures proteins. So they found this mole rat in South Africa that is not fazed by wasabi at any concentrations. It can just eat raw wasabi all day long and no problem. What they found is that it has a very high number of ion channels known as nausin, and these are known to be leaky, making it harder to excite the neurons. So in other words, the mole rat has neurons that are adapted to eating kind of like nasty stuff. Oops, sorry. I gotta keep signed into this in case. I'm multitasking here. Okay. So anyway, when the neurons are leaky like that, they're constantly having to spend energy keeping that electrochemical gradient across their membranes, but they keep leaking, so they're very inefficient. When they took those leaky channels and they blocked them with another drug, then the mole rats became sensitive to wasabi. Okay. So why would the mole rats have this adaptation? Because they're found where ant bites, with these red ants that inject formic acid. And it protects them from the pain of the ant bites. So they can live where other rodents cannot live. So another category are central nervous system stimulants, and they cause an increase in central nervous system activity. Cocaine, caffeine, nicotine. Okay. Anyway, so here's some structures. Again, you don't have to memorize any of these, but you can see why these are in this chapter on amines. Look at caffeine. 1, 2, 3, 4 nitrogen atoms. That's crazy. Tertiary amine, tertiary amine, tertiary amine, secondary amine. So here's nicotine, here's cocaine, here's amphetamine, here's methamphetamine. And what's the difference? They put a methyl group on the amine. So methamphetamine is N-methylamphetamine. That's what it is. Here is dexedrine. Okay. So if you look at dexedrine, look at a similarity to, say, amphetamine. A lot of similarity there. Okay. So that's why I did not want to give this to my youngest son, But when it did, I got the paradoxical effect, because you're simply giving him a stimulant and it caused him to slow down and could actually learn. But a lot of students take Adderall to study. It's really not a good idea. So Socrates was condemned to death in 399 BC for impiety. So impiety was a lack of respect for the gods, which was believed to bring punishment on society because the gods were upset. So essentially, Socrates said to all the other people, I don't believe in all these gods, the gods of rain, the gods of sun, all this kind of stuff. That's all BS. So he refused to go to these ceremonies in their honor of the gods. So they killed him because he refused to go and refused to accept the gods. So they're thinking, oh, he's making the gods mad, so he's going to ruin our lives. So what they did is they gave him hemlock. So hemlock contains coniine. Notice it's a secondary amine, right? There's some stereochemistry here. And coniine causes death by blocking the neuromuscular junction, similar to what curare does. So the muscular paralysis basically makes you stop breathing, and then that's how you die. So if you make the person breathe, put them on a ventilator, you can save that person's life because the coniine will eventually wear off. So here's poison arrow, what do they call it? Poison arrow poison, which is curare. And so it's used in poison arrows. It's from a plant. It's not from frogs. People think it's frogs because they have that frog is the poison arrow frog, but it's not really from them. It's from a plant. And what these two things do is that here's the normal nerve cell, and here is the other nerve cell. So nerve transmission comes this way, releases neurotransmitters, and it's supposed to go over here and cause the effect, right? So this now goes in, sends the signal. But curare and coniine binds to these receptors, acting as antagonists so that the agonist really can't get there and cause its effect. And then we have central nervous system depressants. They suppress central nervous system activity. Sedatives and tranquilizers. So these are barbiturates, antihistamines, marijuana, opium, these kind of things. So here is amobarbital, here is phenobarbital, here is diazepam, also known as Valium. Here's Lunesta. Here's histamine. Here's an antihistamine. Okay. So you're wondering about the structure. Look at the structure of this antihistamine compared to histamine. They don't look similar at all, do they? But the important part is, what are the parts that cause it to bind to the receptor protein? It's all there, otherwise it wouldn't bind to the receptor protein. So here is THC from marijuana. Okay. And we have psychedelics. So here is mescaline. Notice that mescaline is a catecholamine. If you were to take off those methoxy groups right there. Here is LSD. And there's psychedelics. So drugs that affect nerve tissue can either affect neurotransmitters, the receptors, or the ion channels. So what can a drug do? It can enhance the release of neurotransmitters, or it can suppress the release of neurotransmitters. So in other words, I didn't draw the sensory neuron, I just put the rest of the neuron over here. So you have a stimulus that stimulates the sensory neuron. That stimulus could be heat, cold, touch, whatever. That sensory neuron, because of that mechanical or heat-related stimulus, releases a neurotransmitter. But how much it releases can be affected by a drug. The other thing that a drug can do is alter the lifetime of neurotransmitters in the synapse. So here's the neurotransmitters. They're supposed to bind to the receptor proteins, cause a depolarization of membrane, which passes down here and then causes the release of other neurotransmitters over here to go to the next nerve cell. So if the lifetime of these neurotransmitters is shortened or lengthened, you're going to alter the response that this cell right here is experiencing. You're really altering the response, the signaling that's coming from the sensory neuron. There are several very powerful toxins that are designed to stop the destruction, especially of acetylcholine type of neurotransmitters. So neurotransmitters and all biological signals are supposed to be extremely short-lived. It's like this. So if you have an enzyme that normally destroys the neurotransmitters, and then you inhibit that enzyme, then the neurotransmitters won't get destroyed. And then this cell right here is going to get a continuous stimulus that wasn't intended. The other thing you can do is alter the binding of the neurotransmitters to their receptors. You can block the receptors. So if I block these receptors right here, the natural agonists, neurotransmitters, are not going to be able to bind there and depolarize that nerve cell. And then the other thing that you can do is either sensitize or desensitize the voltage sensitive ion channel or ion gate. So what the neurotransmitters do is called a depolarization. So sodium rushes in, potassium rushes out. That's what causes the nerve transmission down its axis. Okay? If that doesn't happen, then you won't release neurotransmitters to the next nerve cell. So let's look at the nerve cell in a very brief manner. There's an enzyme on the nerve cell called the sodium potassium ATPase. It uses energy from ATP to pump 3 sodiums out of the membrane and pump 2 potassiums in to the membrane. That sets up an electrochemical gradient. What is the chemical gradient? Well, sodium is about 10 millimoles concentration in the nerve cell and about 145 millimoles outside. So sodium then wants to rush back in simply because of diffusion. Potassium becomes about 140 millimoles inside, whereas outside is only 5. So potassium wants to rush out. So as soon as I put a hole in there, that's going to happen. And that depolarization travels down the membrane like an electrical wire, gets over here and causes the fusion of these vesicles with this membrane, which empties out the neurotransmitters into the next synapse. So it's a chemical gradient because of the differences in concentrations. It's an electrical gradient because you pumped more positive charge out than you did coming back in. So here's the electrical gradient right here. By the way, how long should those ion channels be open? Isn't it just a very small fraction of a second? It's like this, and then they close up again. So here's one of the things I've worked on before. I studied mitochondria and I looked at the effect of general anesthetics. General anesthetics, what do they do? You've probably been under general anesthesia. They put the mask on you and in a couple of seconds, you're gone. What general anesthetics do is they inhibit the production of ATP by mitochondria. They uncouple the mitochondria. And that lack of ATP reduces the ability of the nerve cells to maintain adequate membrane potential. It doesn't have enough ATP to run the sodium potassium ATPase. So going under general anesthesia is actually very dangerous. That's what has killed several people, even people I know. And that's what killed Michael Jackson. So when you undergo general anesthesia, you need a licensed anesthesiologist right there to monitor all your vital signs. Okay. So I put a bunch of information over here. It's not, it's all in a red box. So I just thought that you might be interested. I was rather interested in it. Some of the things are kind of pertinent. So back in the old days, you could buy cocaine. You could go to the drugstore and buy cocaine. Took the pain away from toothaches and stuff like that. And then what about narcotics, the opiates? If you were to look carefully at this, hopefully you can increase that. And you can see that the United States is just full of drugs. It's really pathetic. I have friends that are very much into guns. I have former police officers, retired police officers, and my son and all of his friends in Waimanalo are all into guns and everything. And anyway, people with guns get really a bad rap because if you look at all the homicides and shootings from guns, who's really doing that? It's not like your average Joe who goes to the firing range with his family on Saturday and then Sunday he goes out and shoots a bunch of people. No, they're criminals with the guns. And most of the criminals, they're either trying to rob you because of a drug addiction, or they're shooting each other because they're competing for drug customers. In other words, they're drug dealers. So if you really want to get rid of gun violence in the United States, the biggest target to me would be get rid of all these drugs. You come across these people, it's like, okay, I'm just a recreational drug user. But, we should really take people's guns away and increase gun regulations. And then at the same time, they don't even know what the gun regulations are. And these are the same people that are giving incentives for profits off of drugs. They're causing it. So addiction is an extremely powerful thing. It's very hard to become unaddicted. Very easy to become addicted. So methadone is an opiate that doesn't give the pleasurable effects. It's effective for some. So we have alternatives to opioids, but whatever. So here's some stuff on Botox. By the way, about 2 years ago, my friend who was his research was being supported by Allergan, decided that they're not going to do any more research on it. We're making so much money, it's ridiculous. And so they're spending their time trying to get the FDA to approve uses for Botox, whether the claims are real or not. That's where they're putting their efforts. It's all about making money. That's why you have these off-label uses and everything else. So here's some funny things about animals. So do animals get high? The answer is yes. And many of them go out of their way to try to get at least alcohol. And yeah. So it turns out that most animals lack enough alcohol dehydrogenase to metabolize ethanol, and so they get intoxicated very easily. And so it turns out that humans, bonobos, chimpanzees, and gorillas have a mutation making their alcohol dehydrogenase 40 times more efficient so that we can actually handle much larger quantities. But I do know people who, if they drink almost anything, they just get totally wasted and to the point where they can't continue. They get just totally sick. So here's something that you should know about, the placebo effect. Basically, you are what you believe. And so one of the things I want to do this coming semester is work with my son and his friends. One of them has a gym and they're all into MMA and everything, and they take all these supplements. So I want to look at the supplements that they're taking, go through it with a couple of friends of mine who are nutritionists and all this kind of stuff, and actually try to tell them whether the stuff is working or not. Because what they do is they hear on the internet, and Instagram is all about making money. There's no facts on Instagram. It's all about selling you stuff. So when you read about this supplement working, you start to believe it. Oh, it worked for these people, so I'm going to take it. Now I feel better. I feel stronger. I feel whatever. And so that's the placebo effect. I have a friend who's since retired, and he taught at medical schools for his entire 50-year career. And this was his field. And he claims that the placebo effect could be up to 50%, depending on circumstances. What that means, if it were 50%, if everybody in a room had a headache, and I gave everybody in that room a sugar pill, placebo, half of the people would say, oh, thank you. That made my headache go away. That's how powerful the placebo effect is. And the nocebo effect is real too. So if you take your kid to get an immunization or something, they're going to get a shot, and you tell the kid, oh my God, this is really going to hurt. The kid's going to freak out, and guess what? It's going to hurt more. So anyway. So any questions at all so far? Okay. I'm going to stop sharing for a minute just so I can look at the chat and don't see anything in the chat. So the allergic reaction is caused by the release of an amine called histamine. So here's what happens. An antigen, such as pollen, for example, reacts with an antibody that you have already. It forms the antigen-antibody complex. And this right here is what goes to tissues and causes the tissues to say, oh my God, we're being damaged, so we're going to release histamine. Histamine is released by cells in an effort to protect the cells or that tissue from something that is attacking it. So then the histamine reacts with receptor cells, receptor proteins on cells. So these are usually called H1 or H2. And that is what gives you the symptoms. So the symptoms are such things like increased heart rate, edema, which is swelling, which is the major thing that you don't want to have happen. Okay, itching, things like that. So where does histamine come from? Histamine comes from the amino acid histidine. Histidine is one of the 20 amino acids you find in proteins. So here's the amino acid, and then you have a decarboxylase enzyme. That would be, I would write this over the arrow right here. And what does it do? It takes off the carboxy group right here. So that goes off as carbon dioxide, and now you're left with histamine. Okay. So why do mosquito bites itch? Well, before we even talk about that, what benefit would it be to you if mosquito bites actually itch? What would be the biological benefit of you getting itchy? You'd learn to swat the mosquitoes away because mosquitoes are vectors for all kinds of diseases, right? But why does the itch actually happen? Because the mosquito injects some of its own saliva, which contains an anticoagulant. The mosquito would have to do that because if the mosquito stuck its proboscis into you and sucked the blood out of you, but it didn't release an anticoagulant, your blood would coagulate right there and the mosquito would be trapped and would die. But those proteins in the anticoagulant and saliva increase an immune response against foreign proteins, and your cells release histamine. Let's see. So here's what happened to me when I was a kid. So I was probably maybe 6 or 7. My father took me to the mainland to see some relatives I had never seen before. And so we get there, and I immediately take their dog, and now I'm riding around in the woods with their dog. And then they start calling me after a couple hours. And so the dog and I come running out of the bushes, and they're totally freaked out because I'm running around in poison oak or poison ivy or something like that. So they thought, oh my God, it's all over you. You're going to be just destroyed by this. And you know what happened to me? 0. Because I had never been exposed to it before. I did not have an antibody made to that antigen. So I could not form the antigen-antibody complex. And so no histamine was released from the tissues. So anyway, later on, we're going to see that in DNA and RNA, the purines and pyrimidine bases are amines. Let's look at purine. 1, 2, 3, 4 nitrogens. Pyrimidine, 2, right? So again, we don't need to memorize these structures, but this is the reason why we really need to have knowledge of all these amines. Okay. So any questions so far? Okay, a couple minutes left. Maybe this is a good point to stop. So we'll just stop here. So tomorrow we'll go through aldehydes and ketones, because it is somewhat like maybe it could be a different chapter. So does anybody have any questions at all?
Not right now.
What about for the exam? You folks know what to do for the exam? So you have 2 hours to take the exam. That means like you asked me for it. I put it in your Dropbox and you got 2 hours to take it and send it back. It doesn't mean that, oh, you have it in your Dropbox and you can just start it anytime you want. No, you have 2 hours once I put it in there, because otherwise I have no other way to time it. So far, I think only one person started it.
How many questions?
Oh, I think it's around 30 or 33, something like that. Yeah. So if I recall, it's a, let me just, I can pull it up. Hold on a second. Maybe I can pull it up. I won't screen share. But yeah, so it's 33 questions. It's, I can't count the pages here. It's not letting me count the pages. Oh yeah, 5 pages. Yep. Nothing tricky. So just tell me what the products are. Name the compound or draw the structure. And there'll be some questions on physical properties. And I think there's one where you have to draw a hydrogen bond between 2 different things. So any other questions about anything? Okay, well, this is going to be recorded. I'll get it up onto the YouTube site as soon as it finishes processing. And you have today to take the exam and tomorrow, right? So anyway, you folks have a great day. And see you tomorrow. And if you have any questions, you can always email. Okay? So take care.
Thank you. Have a good day.
Thank you. See you.